right, good morning, everyone. Welcome to the 27th Annual David Alpers Visiting Professorship, where we have with us today Dr. Timothy Wang. He's the Dorothy and Daniel Silberberg Professor of Medicine, Chief and Vice Chair of Academic Programs in Medicine at Columbia University and Irving Med Center. He's the co-leader of the Tumor Biology and Microenvironment Program at the Herbert Irving Comprehensive Cancer Center, past president of the American Gastroenterological Association, and he does have a connection to WashU. He was summa cum laude at Williams College and AOA at Columbia Medical School before joining us here for internal medicine residency when David Alpers was our chief of GI. He then went on to go to Boston, MGH, and Harvard for GI, where he stayed on faculty before becoming chief of GI at UMass, and then returned to Columbia, where he's had a very long career, was chief of GI there from 2024 to 2025. He's had a long-standing independent career as a researcher with a lab for over 30 years, focusing on relationship between inflammation, stem cells, cancer, and molecular mechanisms of GI carcinogenesis. In recent years, his lab has been known for pioneering cancer neuroscience, having shown that nerves modulate growth of solid tumors. He's authored nearly 400 original publications, extensive reviews, editorial works, books, research collaborations, and grants with exceptional leadership and love of mentoring. He's the recipient of the Arthur and Irene Fishberg Prize for Outstanding Research and the Julius Friedenwald Medal, AGA's highest honor bestowed for outstanding contributions to the field of gastroenterology. So truly an inspiring physician scientist, academic leader, and we're just so delighted to have him back here with us today.
Well, thanks again for the kind introduction, and it's really a great pleasure to come back to WashU, where I did my residency, and be able to give this medical grand rounds. I'm going to be speaking today about gastric disease, both ulcers and cancer, and try to introduce you to the concept of neuroendocrine circuitry and the modulation of stomach disease. So these are my conflicts, disclosures. For the past 10 years, I've served as a consultant for AstraZeneca, GSK, and Novo Nordisk, and probably the work with AstraZeneca is probably of some relevance to what I'm going to be talking about today. But before getting started, I just a bit of reminiscing on the past. This is our medical house staff photo from 1986. There is me on the left here, the point here, down here somewhere, as a senior resident. You know, I don't think I've changed that much, right? But the medical residents at WashU were really great, and I'm sure they still are. And at the time, David Kipnis, our medicine chair, was quite committed in developing physician scientists. I know WashU still has that sort of culture ingrained in it. And over the years, a number of us pursued this sort of career in research, including Steve Shapiro, who's on the photo here, and a couple of residents who are above me who are not, such as Brian Kabilka and Brian Drucker, were really all part of our culture here. Let's see. All right. So let's just go ahead and start. So this is an outline of my talk, and I thought I'd just give a heads up as to where we're going. I'm going to be starting off by talking about peptic ulcer disease, proton pump inhibitors, and gastrin. And I started off actually working in the lab as a fellow on gastrin, and this is something that I've sort of continued over the years. I'll talk about our mouse models of peptic ulcer disease, the role of hypergastrinemia in the response. I'll introduce the gastrin receptor, the cholecystokinin 2 receptor, and its expression on gastric progenitors. I'll describe briefly the role of the vagus nerve in regulating both gastrin responses and ulcer healing. And I'll summarize briefly some of the differences in the role of stem and progenitor cells in healing of ulcer disease, thus protecting against cancer. I'll review work by us on the innervation of the stomach by sensory nerves, the role of these sensory nerves in responding to gastric injury, and I'll focus specifically on the expansion of CGRP, calcitonin gene-related peptides, sensory nerves, in gastric cancer, and finally end with our new model of neural circuits in gastric carcinogenesis and opportunities perhaps for therapeutic intervention. So I'll be speaking on peptic ulcer disease, but I thought I'd start with a brief definition of erosions versus ulcers and address why ulcers are more serious and slower to heal. Both can be caused by the typical agents of H. pylori and non-steroidal anti-inflammatory drugs, NSAIDs, and other damaging agents. But we like to distinguish between erosions, which are smaller, shallow breaks in the mucosa that reach but do not penetrate the muscularis, and ulcers, which are full-thickness breaks that do penetrate the muscularis into the submucosa, and this can erode into blood vessels and even through the serosa and perforate. And the ability of the gastric mucosa to regenerate after injury often determines whether erosions go on to become full-blown ulcers or not. And this concept of a deficiency in regenerative repair in disease causation has now been well studied in numerous organ systems, and I think is key to understanding what's going on here. Now, in terms of treatment of peptic ulcer disease and the history of acid suppression, I thought I'd go back to the very beginning. Karl Schwarz, who in 1904 first came up with the dictum, no acid, no ulcer. And this has been sort of the guiding mantra that acid is sort of the key factor in terms of trying to treat this disease. In the first half of the 20th century, the sippy diet was frequently used to treat peptic ulcer disease, giving milk or cream every 1 to 2 hours to neutralize the acid. It, of course, led to rebound in acid secretion and was not highly effective. After 1940, treatment with antacids became more common, along with the growth of gastric ulcer surgery, Billroth 2, something we used to see 30, 40 years ago, not so much anymore, for recurrent, severe recurrent ulcers. In 1972, Sir James Black, who I got to know working at SmithKline and French, now GSK, developed cimetidine, which was the first targeted drug for acid inhibition, and for which he won the Nobel Prize for the discovery of beta blockers. In the 1980s, H. pylori was discovered by Barry Marshall and Warren as the main cause of peptic ulcer disease, and eradication regimens were introduced. But at the same time, a company, Astra, was developing and testing their new acid-suppressing drug, omeprazole, which was approved in 1989. Over the next several decades, the FDA approved multiple other PPIs, and there's a growth in PPI use, but at the same time, a decline in H. pylori infection and a decline in ulcer surgery. And in 2023, the FDA approved the latest kid in the block, vonoprazan, the first potassium-competitive acid blocker. So from the very beginning, gastrin was sort of part of this whole story of ulcer disease as the main acid-stimulating factor, the main acid secretagogue. It was actually discovered in 1905 by Atkins as an enteral acid-stimulating factor. He called it gastric secretin, or gastrin. It was the second hormone ever discovered. Gregory and Tracy identified gastrin-17 in the 1960s, and Rosalind Yalow in the 1960s also developed the radiohuman assay, and along with Jim McGuigan and others, first measured gastrin-17 and gastrin-34 and solved the ulcerogenic gastrinoma syndrome that at the time was called Zollinger-Ellison syndrome. And that led to the basic idea that gastrin was a major factor driving peptic ulcer disease. At the same time, Rusty Johnson, at the same time, actually first described the fact that gastrin was also a trophic hormone, stimulating proliferation in the GI tract. In the 1980s, it was the molecular period where the gastrin gene was cloned in many different species. Knockout animals were made. And in the 1980s, also, they recognized for the first time that in rats treated with omeprazole developed increases in ECL cell, proliferation of this endocrine cell, and actually developed ECL cell tumors, and this actually delayed the approval of omeprazole for a number of years until it was recognized that this was not really happening in humans. And then in the late 1980s, John Callum actually first described the gastrin link, the fact that in duodenal ulcer patients with H. pylori, they had elevated levels sometimes of gastrin. So just a bit about PPIs. I'll be describing proton pump inhibitors and their effect on gastrin. And just for those of you who don't know the mechanism, this is they bind irreversibly the hydrogen potassium ATPase pump that's actively secreting hydrogen into the lumen. And so they bind to the pump in the secretory canaliculus. Normally these pumps are stored in these tubular vesicles. When you eat, these tubular vesicles fuse with the secretory membrane to form this expanded secretory canaliculus. And when the pump crosses the membranes and becomes protonated, it converts to a reactive thiophilic sulfenamide form that binds covalently to the pump irreversibly. And so you take it really after eating a meal, ideally in the morning. It has a short half-life, but a long biologic half-life. It takes a few days to reach steady state, where 90% of acid is inhibited, and the pH in your stomach rises to 4.5 to 5. But this leads to about 2 to 3-fold increases in gastrin, as I'll describe, about 150 picogram per mil, and then it returns to normal after 3 to 4 days, and that's sort of how these PPIs actually work. And the new kid in the block is this PCAB, this competitive potassium acid blocker, that actually has been around for a long time in Japan, but then the license to this in the US was acquired by a company called Phathom Pharmaceuticals, founded by the late Tachi Yamada, who was a good friend to a number of us. And so it belongs to this class. It's not a prodrug. It has a longer half-life. It actually binds ionically, competing with potassium ions in this hydrogen-potassium exchange. It binds to both active and inactive pumps. It doesn't require a pH to do this. And it's actually a much stronger acid inhibitor and leads to more profound hypergastrinemia, with gastrin levels typically around 500 picograms per mil or even more. So why do you get hypergastrinemia from this acid inhibition? And so this has been years of physiology studying this negative feedback circuit and the fact that acid normally suppresses gastrin through a somatostatin mechanism. And so PPI-induced hypergastrinemia then leads to expansion, a proliferation and expansion of parietal cells and ECL cells. And it leads to this gastric thickening. And even occasionally, as gastrologists know, these benign fundus gland polyps. And around 15 years ago, this became a concern as we were trying to wean patients off of PPIs for a variety of reasons. And they noticed this sort of rebound acid hypersecretion in patients who've been on these drugs for a long period of time. And this was thought to be a major problem, but in the end was solved by basically instituting a slow taper of the medication. But this mild proliferative effect that I'll talk about is reliably seen. So now I'll talk to you a little bit about ulcer healing and wound healing. And so when I was back at MGH, this was a big interest in the different stages of wound healing, mainly in the intestine and colon. And it's also been studied well in the skin. And there's at least 4 stages that are typically required for this healing of a wound, regardless of its location in the mucosa. And so the first step is restitution, where the gastric epithelial cells bordering the ulcer migrate across to sort of cover the damaged barrier. And then there's a recruitment of inflammatory cells that occurs, leukocytes, mainly myeloid cells, macrophages, and neutrophils, and these an expansion of stromal cells that actually then stimulate to some extent this proliferation. And this proliferation is active cell division, typically by transit amplifying type of progenitors, producing a progeny to fill the epithelial defect, and finally, later on, some degree of stem cell activation by slowly dividing gastric stem cells that we and others have studied. And so the model I'll be talking about very briefly in these sort of preclinical studies is an ulcer model. And we've used primarily a number of different models, but the acetic acid-induced model has been very reliable for us in terms of the progression of healing. You apply acetic acid on a little cotton ball that's attached to a pencil-type holder. You apply it to the serosal surface of the stomach for about 60 seconds and then remove it. And this leads to a large gastric ulcer, which then heals over the course of 4 weeks. But it heals pretty quickly, with 30% healing by 7 days, 85% healing by 2 weeks. And we studied in this model also gastric regeneration by a number of other damaging agents to the stomach, agents that sort of kill or destroy cells in the stomach, particularly secretory cells. And these include the H. pylori infection model, or we actually used H. phylis a lot, high-dose tamoxifen, which kills a lot of secretory cells, single-dose, high-dose MNU, which is a carcinogen, and genetic ablation of cells using a diphtheria toxin sort of type genetic model. So we started off studying this gastric ulcer model and examined the responses to injury. And as noted, the ulcers heal pretty quickly over 2 weeks with complete healing by day 28. And you can see the ulcer size here at the top in terms of how quickly it heals. As you can see, by day 14, it's really 90% healed in this study. But we were surprised to see during the normal course of ulcer healing, and these ulcers are primarily in the corpus of the stomach, plasma gastrin levels and gastric G cell numbers were significantly increased, shown here at the bottom. You can see gastrin levels, day 7, 14, 28, G cell numbers, day 7, 14, and 28. There's a rapid 2-fold increase in plasma gastrin levels by day 7, peaking at day 14, and G cell numbers increase more slowly, but also peaked around day 14. And thus it appears, and we've looked at this in other models, that normally during ulcer healing, hypergastronemia occurs as part of this normal ulcer healing process. Now, to address the importance of gastrin cells and gastrin levels in ulcer healing, we decided to generate a conditional gastrin cell ablation model. And so we took the gastrin gene and recombined it and put in this diphtheria toxin receptor, and along with a red fluorescent marker, TD tomato. And this transgenic allowed us to really conditionally delete gastrin cells in the mouse model. And we validated this in our transgenic mouse, showing that if we gave a couple of doses of diphtheria toxin, as shown here, you could really ablate 95% or more of the gastrin cells. And then at the same time showed that this led to a decline in gastrin levels, as shown here, 90% decrease in gastrin levels. And if you followed then after the treatment and saw the regeneration that occurs, then you can see that the gastrin levels then gradually creep up as gastrin cells are restored from the stem cell in the stomach. Gastrin levels became normalized again by about 3 weeks. In order to achieve long-term ablation, we then looked at a model. We gave the diphtheria toxin to ablate the cells once a week, and we could achieve long-term ablation of gastrin cells using this model. So we then looked at the question as to whether the healing of ulcers in the stomach is in fact gastrin dependent. And so we used our gastrin diphtheria toxin receptor ablation model to create this hypogastrinemic state, and then also looked at a hypergastrinemic state, which involved either an Alpers pump giving gastrin infusions over a course of 3 weeks, or we also looked at PPIs. And we talked about if you give omeprazole to an animal, the gastrin levels tend to go up rather quickly as well. And these studies showed that corpus ulcer healing is indeed gastrin dependent. Infusion of gastrin or treatment with a PPI resulted in a marked acceleration of healing with much smaller ulcer sizes at 2 or 3 weeks, as shown here in the bottom of the slide. In addition, ablation of the gastrin cells and reduction in gastrin levels slowed ulcer healing with significantly larger ulcer sizes at 7 and 14 days. Now, it's interesting that this, both the proton pump, which inhibits acid, and which increases acid and lowers pH, and the PPI, which obviously blocks acid and raises gastric pH, had the same effect, suggesting that it may be even more important than inhibiting acid is having the high level of gastrin. And so to address this question as to whether, in fact, it's the gastrin more than the acid that's important to address, we actually we examine the effect of G-cell ablation on the healing of ulcers in the setting of a PPI being given. So as shown here in this model where we combined PPIs and diphtheria toxin ablation of the gastrin cells, G-cell ablation with diphtheria toxin reduced G cells, reduced gastrin, and completely abrogated the benefits of PPIs in ulcer healing. And this is shown at the bottom, particularly in the dark blue in the last lane here, where the PPI results in higher gastrin levels. This is blocked by diphtheria toxin ablation. G-cell numbers were reduced. But the key thing was the ulcer size in the bottom right here, where PPIs led to clearly a strong, inhibition of ulcer formation, but diphtheria toxin completely ablated that effect. So based on these data in mice, PPIs promote ulcer healing predominantly through the induction of hypergastrinemia. So how does hypergastrinemia promote ulcer healing? And the target of gastrin's action is the gastrin receptor, which has been called the CCK-B receptor or the CCK2 receptor, which in the corpus labels mostly ECL cells and a small subset of gastric progenitors high up in the glands, as shown in this cartoon from a recent paper. So hypergastrinemia in these progenitors activates the MAP kinase pathway, stimulates proliferation, and stimulates differentiation towards both the endocrine lineage, this endochromatin-like cell, and the parietal cell lineage. And at the bottom, you can see it also induces histamine release. This is the main role, the physiologic role of gastrin. And when we looked at the effect on progenitors, we tagged our progenitors with this CCK2 Cre-ERT construct. So we were able to genetically trace both the progenitors and the ECL cells that are labeled. And you can see that treatment with a PPI leads to a marked expansion of these progenitors, lineage tracing across the glands. And this is mainly what you see, an expansion of proliferation and tracing with proton pump inhibitors. So we then asked the question about the effect of hypergastrimia on healing of ulcers. And is it it mainly occurs through the expansion of the CCK2R, these gastroreceptor-labeled corpus progenitors. So we basically carried out the same set of experiment-induced ulcers, carried out this lineage tracing of these early isthmus gastric progenitors, and did indeed see that there was a good correlation between the tracing, the progeny generated from these gastroreceptor-labeled progenitors and the healing of ulcers. And as you can see with proton pumps, inhibitors, the PPIs, or with the pump of gastrin infusion, there's a marked increase in tracing across the whole ulcer bed. And if you inhibit this by knocking out gastrin cells, you lose this sort of expansion of the progenitors and you lose healing of the ulcers. So it's been known for many years that in addition to gastrin, the other factor that regulates acid secretion and growth in the stomach is the vagus nerve. And it's another pathway to modulate acid secretion and also particularly a role in healing of the mucosa. So we looked in our ulcer model at vagus nerve fibers that were of the cholinergic type. In other words, nerve fibers that secrete acetylcholine, one of the key parts of the parasympathetic nervous system. And we saw that in response to ulceration, there was a marked increase in nerve stain for this marker VCHAT, which makes acetylcholine, as shown here at the top of the slide. And then we examined the effect of unilateral truncal vagotomy on the gastric mucosa. And so we first looked at it at baseline, no ulcers, just normal mucosa. And by severing the vagal trunk, the anterior vagal trunk, and not the posterior trunk, we could look at effects directly on the stomach mucosa without changing too badly motility and gastric emptying, which is what happens if you do bilateral truncal vagotomy. And also the posterior wall of the stomach could serve as a control for the anterior wall, which was denervated. So this unilateral truncal vagotomy led to thinning of the corpus mucosa, which you can see on the top right of the slide, where you can see the anterior wall is much thinner than the posterior wall. And it also led to a decreased tracing from these progenitors. If you look at the green cells on the left, at the bottom blue panel, the left has fewer of these green cells, so less tracing, less proliferation, and thinning of the mucosa over time. So clearly the vagus, in some ways, modulates proliferation and of the gastric mucosa. And so we then went on to examine the role of the vagus nerve in corpus ulcer healing. And we carried out this unilateral vagotomy model, along with our acetic acid-induced ulcer, with or without gastrin infusions, thinking that maybe gastrin could rescue whatever was happening with the vagus nerve. And so you can see at the, by day 7, we include here both day 7 and day 14 gastrin levels. You can see that cutting the vagus nerve led to a reduction in the gastrin levels, and they actually weren't fully compensated by the gastrin infusion. So there's a marked sustained decrease in gastrin levels with vagotomy. And this led to an increase in ulcer size at day 7, as well as particularly remarkable at day 14, which was rescued only a little bit by adding back gastrin. So cutting the vagus reduces gastrin levels and slows ulcer healing. It slows tracing here as well. And some of the mechanisms appear to be due to gastrin, but some appear to be possibly independent of gastrin. And so the vagus nerve, through cholinergic signaling, communicates with the gastric mucosa through the muscarinic 3 receptor, what's called the M3 receptor. And we could see a similar decrease in gastrin levels and an increase in ulcer size by using an antagonist of the muscarinic 3 receptor called darafenicin. And so we saw that it mimics some of the same effects on gastrin, on the ulcer size, on lineage tracing as shown here. And also it had direct effects, I think, on the corpus progenitors. We could show using these sort of 3D organoid culture that you could again suppress the growth of organoids as shown here in this quantitation at the bottom by this M3 receptor antagonist. And you could rescue it to some extent by an agonist, which was pilocarpine. So somewhat of an unexpected finding or surprise to us was that hypergastrinemia appeared to protect against the development of gastric dysplasia and cancer in 2 different models, an H. pylori model and a MNU, a nitrosamine carcinogen model. And so thus, when we ablated G cells, gastrin cells with diphtheria toxin, it led, as you can see in this top here, worse dysplasia, while gastrin infusion suppressed dysplasia. So the G cell ablation in combination with H. pylori infection led to actually greater colonization, greater inflammation, atrophy, metaplasia. You can see the increase in H. pylori colonization by this PCR for mRNA for H. pylori. And this was then suppressed by either infusion of gastrin through a Alzette pump or through, in the combination actually of a pump actually and H. pylori actually, again, reduced inflammation and reduced dysplasia. So, and one of the interesting things is why should this be when it's in fact stimulating progenitors? So in fact, again, we confirmed that we, in the H. pylori model, when we ablated gastrin cells, we saw less lineage tracing. And so through other studies that I don't have time to show you, we concluded that the beneficial effects of gastrin in promoting the healing of ulcers and expanding these progenitors actually protects against cancer. As we've been able to show now in other studies that the cancer and the dysplasia cells actually arise from stem cells. So by stimulating proliferation of progenitors, it reduces the need for activation of these more quiescent stem cells that we believe are the main origin of cancer. So we have some data, I guess, from our clinical studies from patients that we can also see in pre-neoplastic conditions, particularly patients with multifocal atrophic gastritis, as defined by pleochorea, which is the worst precursor to gastric cancer, that these patients actually have lower levels of gastrin cells, endocrine cells in the antrum of the stomach, in the setting of this atrophy. And in fact, antral atrophy, one of the definitions really is a reduction in these secretory endocrine cells, these G cells. And so we saw that was the case in patients with atrophy, in contrast to non-atrophic patients or patients that had corpus ulcers, which in fact had elevated levels of gastrin cells. Again, suggesting that this loss of G cells may be really a biomarker or perhaps even a partly causative factor in the development of gastric cancer. So this was a summary of our model, which is that hypergastrinemia, whether through physiologic or pharmacologic stimulation, induces these CCK2R, these gastrin receptor progenitors that accelerate corpus ulcer healing and perhaps reduce gastric dysplasia. And that vagal signaling also plays a role potentially through the muscarinic 3 receptors through some sort of a neural circuit. And one of the things we're still studying is actually how these neural circuits get activated and the relation of gastrin cells and secretion of gastrin to actually sensory nerve signaling within the gastric mucosa. And while the relevance of these findings to patients is not completely settled, we do suggest that this hypergastrinemia is integral to gastric ulcer healing. Gastrin levels might be a good biomarker and should require further study in that setting. So now I'll switch gears a little bit and talk a little bit more about our neuroscience interests. And I'll start off by just talking a little bit about the extrinsic innervation of the stomach. So I won't really be talking as much about the enteric nervous system, but more on the nerves from the extrinsic system that innervate the stomach, particularly the vagus nerve. So the vagus nerve comprises both parasympathetic fibers, as I talked about, and I showed you with VCHAT staining, that secrete acetylcholine, and they originate in the medulla oblongata in the brainstem. But they're also very, very abundant, in fact, more so than cholinergic fibers, sensory fibers, which originate in the nodose ganglia in the neck, and sense aspects of the GI tract, as well as secrete a variety of neuropeptides. And so we've been studying or defining in more detail the origin of these sensory nerves that innervate the normal stomach. They also come as well from the dorsal root ganglia, shown here in this little slide at the bottom, this ganglia adjacent to the spine through most of the thoracic and lumbar spine. And so we carried out retrograde tracing of the normal stomach using 3 different tracers shown here, FAST Blue, CTB, Lexiflor, and EAV, HSYN EGFP, which we injected into either the anterior or posterior wall of the stomach, and then looked for the tracer in the relevant ganglia, the nodose ganglia in the neck, and the dorsal root ganglia adjacent to the spine. And we found consistent expression of these tracers in the ipsilateral nodose, or DRG, and the DRG, as shown here, is from between T7 and T13. And immunostaining of these nerves showed that they actually there was innervation from both classes of sensory nerves, both peptidergic nerves, as well as non-peptidergic nerves, which could be analyzed with IB4, isolectin B4. But the vast majority of these nerves were of the peptidergic type, and the vast majority stained for this particular peptide, CGRP, calcitonin gene-related peptide. And so we then looked at whether these CGRP-positive sensory nerves, which again come from the nodus ganglia down through the vagus nerve and are sort of ablated to some extent with vagotomy, whether they played an important role as well in mediating gastric ulcer healing. And so we did a number, again, of our acute gastric injury models, including the ulcer model as well as other different models, and looked at what happened first with those nerves, and we found that they increased. These sensory nerves also increased with ulcer healing. And we found the reason for their increase to some extent was the upregulation of a neurotrophin that we've studied called an NGF, nerve growth factor. So in response to any kind of gastric damage or gastric ulceration, you see an upregulation of NGF, both in some subsets of epithelial cells as well as in some stromal cells. And this NGF is what leads to the expansion of sensory nerves in the gastric mucosa during ulcer healing. And so then we asked whether we could test the functional significance of these sensory nerves. And here we used what's called a DREAD receptor, a designer drug-related receptor. This was HM3DQ, as shown here, that we put into sensory nerves using the TRPV1 targeting vector. And this is a modified version of the human muscarinic 3 receptor used by a lot of labs and can be activated by drug clozapine N oxide or CNO. And so you put this into the sensory nerves, this aberrant receptor, activate it with a special drug, and you can cause activation of these sensory nerves. And when we did that, we saw increased release of CGRP. We saw increased proliferation. If you gave it to mice for 40 weeks, this designer drug, they developed marked gastric hypertrophy and hyperplasia of the cells with all this Ki67. And so we could actually induce proliferation through the sensory nerves in the stomach. And then we looked at our ulcer model. We created our acetic acid-induced ulcer model and then stimulated the nerves for 10 days with the drug and saw increase in proliferation again and a much faster ulcer healing. So these sensory nerves clearly can play a role in mediating regeneration or healing responses, and consistent with the study I showed you where vagotomy ablates to some extent healing. And it raised a number of questions about ulcers and ulcer healing and the wound that does not heal, which of course is cancer, and how these nerves could play a role actually potentially in cancer formation. And just in terms of the receptor for these, for CGRP is actually a heterodimer between the calcitonin-like receptor, CAL, CRL, and RAMP1. And RAMP1 has been studied by a number of labs. And here we perform in situ hybridization in the normal stomach looking for RAMP1 expression. And interestingly, there's 2 hotspots for RAMP1 expression. One was actually in chief cells here at the bottom of the glands, and the other was in the isthmus of the stomach, just in the exact location of the progenitor zone. And we've been looking at co-localizing this location. And we were able to show that this is a stem or progenitor cell through 3D organoids where we actually stimulated the growth of gastric organoids using CGRP treatment, suggesting that it is expressed in a subset of these stem progenitor cells in the isthmus of the stomach. And so as I talked about, cancer is the ulcer, the wound that will not heal. And so we began thinking about the role of nerves in cancer now about 15 to 17 years ago. I was part of a NCI program called the Tumor Microenvironment Network. And we're mainly looking at the tumor microenvironment, thinking about the roles of fibroblasts and macrophages and other stromal cells. But several of us, Gustavo Ayala at Texas, and I became interested in nerves. And so we started working on these in the area of particularly prostate cancer, which several groups worked on, and stomach cancer, which was my interest back in that time. And then the first paper started to come out. Paul Frenette, the late Paul Frenette, came out with the first paper in Science in 2013, and we came out with our paper on stomach cancer a year later. And this led to actually an NCI workshop in 2015 that I attended. Paul Frenette helped organize, and another investigator, Michelle Manji, was part of this workshop. And she was studying the relation between nerves and cancer in the brain. And so then in 2019, 2 really landmark papers came out by Michelle Manji in one group and Frank Winkler in the other, showing that brain cancers form direct electrical synapses with neurons and form part of this overall synapse. And the cancer also remodels the neural circuits within the brain. And then that led to a Banbury conference that we held in 2019, where we finally decided on naming this field cancer neuroscience. And this led eventually to this Cell paper by a number of us in 2023, with the idea that somehow the entire nervous system and even the brain are involved in interactions with cancer cells, and that this leads to systemic effects on nerve interactions throughout the body, including nerve interactions with immune cells that may be modulating immune suppression in the setting of cancer, as well as mental effects that you can see sometimes with cancer. And so this became a very interesting area for us, and a lot of our lab is now focused on this. But we started here with gastric cancer, and fifth most common cause of cancer worldwide, with around 800,000 deaths in 2020. It's defined by the NCI as one of the recalcitrant cancers with a 5-year survival rate of under 50%, strongly linked to H. pylori infection, the major risk factor. Interestingly, there's a rising rates in gastric cancer in the US in patients under the age of 40, similar to colon cancer. And many of these patients with gastric cancer don't have H. pylori, so there are other things going on with gastric cancer besides H. pylori. I became interested in this actually in medical school and residency, and during residency pursued several studies and papers with the late Ray Klaus and Catherine DeSchryver, and these are referenced here. We led to 3 different papers suggesting that this was foreshadowing my later interest in gastric cancer in mice, which of course is also important. So we began in around 15 years ago, based on our discussions from the tumor microenvironment meeting, to carry out surgical vagotomy in mouse models of cancer. And we again carried out both unilateral and bilateral vagotomy. And we found that vagal denervation suppressed cancer growth. And when we did it unilaterally, it was only on the denervated side of the stomach. So if in the mouse model, the posterior stomach, the cancer grew fine, suggesting a very, very direct effect. And we confirmed that VCHAP-positive cholinergic nerves were increased in gastric cancer models, that the release of acetylcholine led to upregulation of Wnt or Hippo signaling, that it was, again, related to NGF expression. And so we really established this feed-forward mechanism where the cancer brought in the nerves, and the nerves then stimulated the cancer, and they made more NGF and brought in more nerves. And so at this time, though, we're focused really only on cholinergic nerves, based on our knowledge of gastric physiology, and we're not considering adrenergic nerves or sensory nerves. So we went back and tried to characterize all the different types of nerves in the stomach in these gastric cancer models. And this is an immunohistochemical study of 4 different gastric cancer models. Actually, just in the slide, I show 3 of them. An MNU carcinogen model, this is a nitrosamine model, a KRAS model, and then a triple transgenic KRAS P53CDH1 knockout model. And we looked at the abundance of the different nerve types in all these different models. And as you can see, we have controls alternating with the cancer models in this slide. As you can see at baseline, cholinergic nerves are predominant. We sort of know this, that it's highly cholinergically regulated, not so much for the sympathetic nervous system with tyrosine hydroxylase. But in response to cancer, the biggest increases by far were in sensory nerves that all stained positive for CGRP. And we confirmed this in human cancers as well, in these tissue tumor microarrays staining for CGRP were markedly increased in both intestinal and diffuse cancer. We showed in our mouse models that the receptor RAMP1 correlated with the expansion of dysplasia in this triple transgenic mouse model. And we also did bulk RNA sequencing and looked at correlation with survival in those cancers with the highest levels of RAMP1 expression. The CGRP receptor, as shown here in red, had the worst survival, suggesting this may be clinically relevant. And we also did a retrograde tracing of our gastric cancer model here, a syngeneic orthotopic ACKP gastric cancer that was lineage traced with a delta G deficient rabies virus. And again, we saw labeling in nodose ganglia and the dorsal root ganglia. And all of the nerves that were traced here from the cancer were positive for CGRP and at higher levels of the NGF receptor, which is called TRAC-A, and this was verified by flow sorting. So again, going back to our different models of modulating these sensory nerves, we examined the effect of activation of sensory nerves in the gastric cancer models using our chemogenetic model, our HM3DQ modified muscarinic 3 receptor activated by the DREAD agonist CNO. And we showed in both the MNU carcinogenic model as well as this orthotopic syngeneic ACKP model, where this cancer cell is just implanted into the mucosa of the stomach, and showed that activating the sensory nerves in both cases stimulated growth significantly, led to decreased survival. And because these sensory nerves secrete CGRP, we actually addressed the question of what happens if you inhibit the CGRP receptor. And in fact, there's a number of drugs that block CGRP signaling, and they're migraine drugs. They're called GPANs. And so here we used one of these, which is rimegepant, and showed that by blocking CGRP, you could extend survival in these mouse models of cancer. And again, ablation of these sensory nerves also attenuated gastric cancer progression. And we did this two ways. Shown here is this work we're using genetic ablation, putting in the diphtheria toxin receptor and treating the mice with diphtheria toxin, which ablates the sensory nerve, the TRPV1 sensory nerves. And this slide shows that we could successfully ablate most of the sensory nerves in both the JNC, which is the nodose ganglia, and the DRG. And then this led to decreased growth, both in the MNU carcinogenic model as well as the orthotopic model, significant decrease in growth. And we got the same result using another agent, resiniferin toxin, which is analog of capsaicin, that is cytotoxic to TRPV1 sensory nerves, also inhibited the growth of gastric cancer cells. Now, what I've showed you mostly so far is the effect of sensory neurons on early tumor growth. And we wondered whether sensory nerves could also play a role in more advanced stages, invasion and metastasis. So we set up initially an in vitro co-culture model, as shown here in the top left, where the nerves are labeled with red, the cancer cells are labeled in yellow. I'm not sure how well this projects. But in response to, and these sensory nerves carried the DREAD receptor, when we treated them with the agonist CNO, the cancer cells actually migrated towards the sensory nerves and lined up right here all together with the sensory nerves. So clearly it can induce the migration of cancer cells. Then we looked at our orthotopic model and said, well, what about invasion, for example, into the portal vein? And so here, these FACS plots simply show FACS of the cells in the portal vein for the cancer cell marker. And so in response to chemogenetic activation, we saw a huge increase in these, we call circulating tumor cells, CTCs, in the portal vein. And finally, we used a metastatic model where we implanted the gastric cancer cells into the stomachs of the mice, waited 2 or 3 weeks, 4 weeks, and then carried out gastrectomy and esophagojejunostomy in the mouse model. And these cells were labeled with luciferase so we could do bioluminescence imaging as well as later histologic evaluation. And what we saw overall, if you look at the bottom, the increase in bioluminescence increased about 4-fold, and we confirmed the increased presence of METs to both the lymph nodes and the liver following chemogenetic activation. So sensory nerve activation can clearly appear to drive metastasis as well. Now, we're very interested in whether the cancer cells and the nerves were interacting directly. And so I'm just going to show you a series of set of images with most of the nerves are labeled sensory nerves. These are DRG nerves in red, and the cancer cells are in yellow. And we looked at co-culture and then activation of them with a chemogenetic model of a CNO treatment. And so what you see is normally the cancer cells do show these occasional interactions with the nerves. They have very close apposition of nerves to the cancer cells. And a big question in the field is whether these are really true synapses or not, and that's been still being debated in the outside of the brain. But in response to CNO treatment, you see an increase in the interactions, much more intense interactions with cells migrating towards the nerves also showing some outgrowth of axonal fibers. And this could, again, be blocked by rimegepant. So CGRP is clearly mediating, to some extent, these interactions between the cancer cells and nerves. And we actually went out to isolate the cancer cells that directly interacted with the nerves and carried out RNA sequencing, comparing them to cancer cells that showed no neuronal interactions. And the cancer cells that interacted with the nerves showed an upregulation of neuronal transcription factors, NeuroD1 and ASCL1. And the nerves that interacted with the cancer cells showed upregulation at their tips of synaptophysin, which tends to mark synapses. So this really does suggest that neural activation drives this interaction between cancer cells and nerves and results in these like synapse-like connections. We wondered if the cancer cells were talking to the nerve, so we actually screened a number of different possible receptors using this technique called GRAB, G-protein coupled activation-based neurotransmitter sensors, which we separately infected the nerves with, and then looked at which nerves could be activated by cancer cells. And for the cancer cells, we put in a channel rhodopsin, and so we could activate the cancer cells with optogenetics. And so what we found, the bottom line, was that glutamate sensors and adenosine sensors could lead to these sort of signals. You can see these yellow signals in the nerves in response to cancer cell signaling, suggesting the cancer cell is, in fact, talking to the nerves as well. And finally, we just wanted to confirm again this interaction between cancer cells and nerves. And to do this, we put calcium sensors, which is a form of a measure of sort of depolarization, first into the cancer cells and then into the nerves. And so on the left is a panel of images where the cancer cells express this calcium sensor, JR gecko, which is a DS red fluorescent marker. And so when we activated the nerves using our chemogenetic activation, we could see calcium signaling, calcium flux, in the cancer cells over 1 to 10 minutes, suggesting a slow release of internal calcium rather than voltage-gated calcium channels. And this is consistent with what's known about RAMP1 signaling, that it can lead to this sort of internal release of calcium in response to signaling. And on the other hand, when we put the cancer cell into the stomach of mice and put the calcium receptor, here we used GCAMP6S, into sensory nerves, we activated the cancer cells with optogenetic activation. They expressed the channel rhodopsin. We could see over 1 to 2 seconds, I don't know if this shows up very well, this red color appearing here in the nodose ganglia. And again, that shows that the cancer cell can actually lead to depolarization of the sensory nerves. So how does CGRP or RAMP1 signaling lead to cancer cell growth, and does it do it directly to some extent? And so we actually carried out bulk RNA sequencing of our cancer cells with and without sensory nerves, with or without sensory nerve activation by CNO, did a gene set enrichment analysis. And in almost every comparison we looked at, the signaling pathway that came up was RB, E2F. E2F is an activator that when RB is inactivated. And this was also through a signaling pathway that involved PI3 kinase and CAM kinase. And we confirmed that this RB pathway was likely relevant to CGRP signaling by staining of human gastric cancer samples with phospho-RB. And so phospho-RB signaling correlated with CGRP signaling in our gastric cancer patient samples. So what I've told you about so far is that gastric cancer can form these synapse-like connection with nerve terminals, with sensory nerves. And this is based on the finding that there's an extremely close juxtaposition of cancer cells and nerves. We see upregulation of neuronal transcription factor and gastric cancer cells that are connected to sensory nerves. We see upregulation of saprotophysin in these connected nerves. And we've been able to show bidirectional communication and functional electrical connection, including, as shown here, we've carried out some single-cell nerve patch clamping between gastric cancer cells and nerves. And so we believe that this feed-forward circuit can be initiated by signaling either from gastric cancer cells or from nociceptive nerves as well. Now, the one gap in our evidence that we've been unable to achieve has been getting good electron micrograph, transmission electron micrograph, EM images. But this gap has since been filled since we published this paper by several other groups. And in particular, studies in lung cancer, 2 papers in Nature by colleagues looking at non-small cell lung cancer, at small cell lung cancer, where they're able to show clear synapses between nerves and cancer cells, particularly in metastases to the brain. In the lung itself, the EM images, I think, are still not as clearly clear-cut. And in pancreatic cancer, a recent publication just this past month in A Cancer Cell by Amir Dakin, and we wrote this editorial about this in this preview. He defined this as a functional pseudosynapse, which I think is also a reasonable term. But the idea that there's direct connections between cancer cells and nerves are probably playing a major role in cancer cell biology and progression. So the major finding in our study is that gastric cancer and peptidergic neurons form this sort of complete neuronal circuit. And we've shown that there's bidirectional communication between the peripheral nerve ganglia, nodus ganglia, and DRG, and the cancer cells. The cancer cells can signal to the nerves through things such as glutamate and possibly adenosine. The nerves clearly can signal, sensory nerves, through to the cancer cell through CGRP. There may be other neuropeptides. There's been a lot of interest in glutamate as well as a signal from the nerves through NMDA receptors in extracranial tumors. But this neural circuit, once it's activated, can, and we've been able to show now changes in activation in nodus ganglia, increased CFOS and ERK staining and so on, along with calcium staining, can lead to signals elsewhere. So one thing we know is that these electrical signals can tend to travel. And what we've been able to show in the lab in the last year or so is that this leads to effects on fibroblastic cells, stromal cells. There are connections between sensory nerves and fibroblasts. And this can lead to the development of CAFs. It can lead to changes in immune cells. And that's been shown by other groups that T cells can be led to exhaustion by CGRP signaling, and we've been able to confirm that as well. And it can lead to signals to the brain. And so now many groups are now looking at activation of specific areas of the brain in response to cancer signals coming through the sensory nervous system. And so the major question is whether any of these can be targeted therapeutically. I've talked to you a little bit about CGRP inhibitors such as rimegepant and atogepant. There's ways to block neurotrophins that create these interactions, TRAC inhibitors. This can lead to activation of the sympathetic nervous system, and so many groups are now looking at beta blockers and other drugs. So the hope is whether or not in the future we can somehow unhook this connection between the nervous system and a cancer and lead to some sort of increased, better clinical therapeutic intervention in many different types of solid tumors. So I'll stop now and just thank members of my laboratory who've been involved in this, and happy to take questions. Thank you.
Thank you so much. That was wonderful, Tim. So I actually had a couple of questions for you. What's known about human variants in these receptors, CGRP and glutamate receptors, and maybe effects on metastasis?
That's a good question. I'm not sure. Glutamate receptors, yes. There's been a lot of studies, particularly in the CNS system. But I don't know anything really about variants in CGRP receptor. It's a really good question, though, and that would be potentially another biomarker for patients at risk for cancer, absolutely.
Yeah. Actually, we probably should get the
last...
the most important slide.
Yeah,
the other question I had is, I wonder if you considered, these newer variants of GLP-1s, many of which have effects on calcitonin receptor. And there's a lot of debate, as you know, in terms of, how GLP-1s are so effective independent of weight loss. And the possibility that there are receptor agonism effects on cancer incidence and mortality, GLP-1s as a therapeutic target for cancer.
Yeah, and that's a great question. In fact, I sent a note to one of my postdocs a month ago telling her to work on this GLP-1 angle because sensory nerves have receptors for GLP-1, and signals from the sensory nerves can go into the brain. And it's been known for a long time that GLP-1 has central effects, and people have been wondering whether it's simply through circulating hormone or whether there could be other ways of sensing. I should also mention that I'm involved in a collaboration right now about the role of the sensory nerve pathway in cachexia. And so one of the things that happens during cancer often is a huge amount of weight loss. And there's good evidence now that this weight loss is in fact partly mediated now through a neural pathway. So I think the combination of that and the GLP-1 angle and the fact that GLP-1 receptors are present on sensory nerves and haven't really been studied extensively is a really good point.
Thanks. Jose?
Wonderful topic. I just have a question about the context of hypogastria. So in your mice, do they develop ECL hypoplastic lesions or neuroendocrine tumors. Do you think that in the context of ulcer healing, maybe you're skewing away from a hyperplastic response on the ECLs and instead skewing towards ulcer healing? Because as you know, in humans, for example, with autoimmune gastritis, high gastric levels are presumed to drive these
Just repeat the question.
Yeah, the question really relates to hypergastrinemia and the effect on ulcer healing and the effect on ECL cells, and whether in the mice, many mouse models actually develop ECL hyperplasia and or ever ECL cell carcinoid tumors. So just a bit of history. These carcinoid tumors were really described in rats. There was actually a fair amount of investigation of PPIs and so on mouse models. Mice, it turns out, are much more resistant to ECL cell tumors, more similar to humans in that sense. They do develop ECL cell hyperplasia, but they don't develop those sort of nests of tumors that sometimes you see in rats. And the other species is another type of rat. The mastomus is another model that really develops ECL tumors in response to high gastrogens. But most other animal models don't. It's not clear why. No one really has a good model, I think, yet for ECL carcinoids in mice, although I know Juanita Merchant has been working on this to some extent. But the question of whether or not the ulcer healing versus ECL cell hyperplasia is sort of related or even inversely related is a good one. I guess the bottom line is whether or not this is relevant to human patients in terms of ulcer healing, and that's something we don't really have much data on yet. We can hypothesize, but it seems logical that it would play some role. How important, if it's as important as it is in mice, I don't know. I can't say for sure. Thanks.
Jeff?
Yeah, also I'll reiterate the fantastic talk. So what I was wondering is, if you look at another intestinal metaplasia, Barrett's, does it express CCK2R? So is the meprazole effect, like reducing dysplasia in Barrett's, actually just due to gastrin, or do you think it's due to something else?
Yeah, so the question really relates to PPIs and the effects on Barrett's esophagus. We know that for GERD patients with Barrett's, PPI treatment is standard therapy, and it has been shown to sort of promote healing. And whether or not it's simply due to acid reduction and acid injury to the esophagus, which is certainly part of the effect, or whether the hypergastremia could actually be playing a somewhat protective role there as well. I will say that, yes, CCK2, CCKB receptors have been shown to be expressed in Barrett's, which is includes a lot of gastric-like metaplasia, and so expresses the gastroreceptor in Barrett's. But no one has really looked at, and it's a good question. In our mouse models, we have shown that that can be lineage traced by hypergastrinemia. Now, whether or not gastrin promotes more cancer so far, it still looks like it protects against cancer. And so I guess according to what you're just suggesting, that probably may be a very similar role, with expanding progenitors in Barrett's, thereby reducing, for example, activation of a cancer-type stem cell. So that would be our hypothesis. Yeah, thank you.
Thanks for a really nice talk. With respect to gastrin and ulcer healing, do you think that we should start looking into the gastrin agonists to treat acute GI bleeding?
Yeah, so the question was, since we've been able to show that hypergastremia in the mouse model can promote ulcer healing, and PPIs are effective partly because of that, would it make sense to actually consider a gastrin agonist to actually stimulate the gastrin receptor in the setting of really bad ulcer disease? I don't know. It's something to think about. I mean, but proton pump inhibitors and other drugs right now raise gastrin levels pretty well. Would a gastrin agonist on top of that have much of an effect? And again, in patients with severe GI bleeding, the issues, the healing is almost secondary to treating the bleeding. So I don't think it would make, there's a really need for that, but it's something to think about. It's something, if we had an agonist, we'd certainly love to try it in our models. Thank you.
Thanks a lot, Tim. That was great. Okay. Terrific.
Thank you.
A lot to think about.